And then without further ado, first speaker to come up is going to be Alicia Harrison from END at CHOP, and she's going to talk to us about the ProMMiS Natural History Study. So ProMMiS is the US-based prospective natural history studies for SYNGAP1 related disorders. Alicia, I'm filling the time a little bit while you're walking up here. Yeah? And Alicia is our lead genetic counselor for SYNGAP1.
Should we use this to advance the slides? All right, so hey everybody. So my name is Alicia Harrison. I am one of the genetic counselors at the Children's Hospital of Philadelphia. And I'm going to be giving some updates on the SYNGAP1 ProMMiS Natural History Study. So just a quick overview. We're going to go quickly over the protocol design as well as some recruitment updates, and then because the study's now been going on for 2.5 years, we're going to start giving an overview of some of the natural history data that's been collected, and then we'll also talk a little bit about some of our more exploratory work. So to quickly review the protocol and study design, the SYNGAP1 ProMMiS study is the first prospective multi-site, multidisciplinary natural history study for SYNGAP1 related disorder. And it was sort of originally conceived as having two distinct phases. The first year of the study was the cross-sectional phase of the natural history study, so that was intended to recruit as many individuals as possible with SYNGAP1 related disorder to get kind of a broad overview of what the phenotype looks like. And after that first year, we transitioned into the longitudinal phase of the study, where we ask families to come back every 6 months so that we can collect data about how individuals' phenotypes change and evolve over time. When a family comes to the natural history study, they come for the entire day. They get a sort of whole battery of different clinical outcomes assessments. They see developmental pediatrics, neurology, genetics, as well as speech, occupational and physical therapy, and they get a sort of whole list of assessments, which you can see up there on the screen, to take a broad look at their development across multiple domains. So this slide is for the compliance folks in the room. The ProMMiS Natural History Study is structured as a consortium, so individual data is collected at each one of the clinical sites, and then funneled to the Data Coordinating Center at Children's Hospital of Philadelphia, where it can be compiled and analyzed and put together so that it can be used by industry partners, advocacy organizations, and other researchers. And while the natural history study itself is not a clinical trial, the data within this natural history study is being collected in a GCP compliant manner, which is to say that it has the same amount of monitoring and standardization that would be required for clinical trial data, and that hopefully aligns the study with FDA regulations so that the data collected within the natural history study could be used in future clinical trials. So switching gears a little bit, we're going to talk about recruitment. So the original goal in terms of recruitment for the natural history study was to recruit 100 people with SYNGAP1 related disorder. We're already well over that goal. We have 117 individuals that have been evaluated within the natural history study. You can see that's the blue line there on the graph, and then the yellow line represents the total number of visits. So you can see that now that we're in the longitudinal phase of the study, we have many patients coming back for their 4th and 5th follow-ups, and we're starting to get some really nice longitudinal data. And those 117 individuals are about 50-50 male and female, and span sort of the whole age range, ranging from less than a year old to older than 68. But the majority of people within the natural history study are between the ages of 2 and 12, so majority are school-age children. People do come to us from all over the United States, so you can see people are coming from all over the country, as well as some individuals from outside of the country, like Canada, Argentina, and the UK. And all of these individuals are seen at one of 3 different clinical sites. So the original site is at Children's Hospital of Philadelphia, but we also have sites at Colorado Children's with Megan Abbott's team, and at Stanford with Juliette Knowles' team. So to transition now a little bit into talking about the data that's been generated by the natural history study, we're going to first talk about epilepsy histories. So we collect data at each natural history study visit about an individual's seizure types and seizure frequencies, and we use that data to retrospectively reconstruct what epilepsy histories look like for people with SYNGAP1 related disorder over their lifetime. And this is a technique that has been sort of developed over the last 5 or 6 years by Ingo Helbig's lab with support from several federal grants. So to orient you to this figure, this is each row in this figure represents an individual with SYNGAP, and each dot represents seizure frequency in 1 month time bins for those individuals. And we know that SYNGAP1 has a unique epilepsy profile, even relative to other developmental and epileptic encephalopathies. And you can see it here compared to STXBP1, which is another condition that we do natural history work on. And seizures in SYNGAP tend to start a little bit later than some other conditions, but once they do onset, they can be very explosive, and we can see a very high number of seizures per day. And we think that overall, about 80% of individuals who have SYNGAP1 related disorder have epilepsy at some point in their lifetime. And at any given moment in time, about 50% of individuals with SYNGAP are actively having seizures, with the majority of those individuals who are actively having seizures having several or many seizures per day. And we can break this down more specifically as well by seizure types. So these are the most common seizure types seen in SYNGAP: myoclonic seizures, atonic seizures, and absence seizures. And I think it's worth noting in this analysis that we think only about 10% of individuals with SYNGAP have generalized tonic-clonic seizures. So the predominant seizure types that we see in SYNGAP are not necessarily countable motor seizures. And this is going to be important going forward because it's going to inform the way that we think about seizures as a potential endpoint for clinical trials. And then switching gears a little bit to talk about the developmental scales. So we do a number of different developmental assessments at every single natural history study. One of them is the Bayley-4. This is a norm reference test that's designed for children between 0 and 42 months of age. But we do this test for everybody who comes through the natural history study regardless of their chronological age. And it's meant to assess development across a number of different domains: cognitive, expressive and receptive language, and fine and gross motor skills. And here we're looking at the gross motor and fine motor subscales of the Bayley, and we have seen actually a really nice, clean progression over time with the data from the motor scales of the Bayley. So you can see a nice developmental trajectory over time. And this is a little bit in contrast to some of the other subscales of the Bayley, like this is the receptive communication and expressive communication subscales, where we see a lot more variability in that data, which is a little bit similar as well to the cognitive subset section of the Bayley, where we see a fair amount of variability as well. And we attribute some of this to behavior. I don't need to tell anybody who's in this room that behaviors in SYNGAP1 can be very challenging, and on things like the Bayley, to some extent we think this variability reflects not so much developmental variability as variability in sort of participation and cooperation and how much an individual is willing to participate in the assessment on any given day. We also do the GMFM, which is the gross motor function measure, which was originally designed to look at gross motor function in children with cerebral palsy. It's not a norm reference test, so in this test, children are compared to themselves over time. And it's designed to sort of max out at the skills of a typically developing 5-year-old. And here again, sort of similar to the gross motor and fine motor subscales of the Bayley, we see a really nice, clean developmental progression. So we're seeing some good and promising data out of that as well. And then the last one is the Peabody-3, which we use the fine motor subtests of the Peabody. Because we want to evaluate hand manipulation and eye-hand coordination, which are things that we know can be challenges for people with SYNGAP1 related disorder. So here too we see a nice developmental progression and see those scores changing over time. So while seizure histories and developmental histories certainly are kind of the core of the natural history study protocol, we do also have a number of exploratory projects as well. So the first one of those is quantitative EEG. We do a quantitative EEG on every individual who comes through the natural history study. I'm not going to talk about that a lot in this talk just because we have a whole separate talk later in the day by Ian McSally who's going to go over quantitative EEG, so I'll leave that to him. We also collect data using motion sensors at every natural history study visit. And we're just now starting to send individuals home with watches that assess sleep actigraphy to assess sleep at home, and then those watches can be sent back to the study team. So all of these are sort of exploratory projects that are still in the works. We did also, in the course of doing the natural history study, identify several familial cases of SYNGAP1. And we noticed that a sort of preponderance of those familial cases, or cases where those variants were inherited from an affected parent, were kind of clustered at the beginning of the gene with variants in the first 4 exons. So this has also led us to do a lot of work on genotype-phenotype correlations in SYNGAP1. But Jill McKee does have a talk on this later in the day, so I'm gonna leave that to her to answer questions on that. So to summarize everything, this was updates from the ProMMiS Natural History Study, which is a prospective multidisciplinary natural history study for Syngap that's been running for about 2.5 years. We've seen really good recruitment over time, and we're starting to get some nice longitudinal natural history data that lets us look at seizures over time as well as development over time. And then we also have a fair amount of exploratory work looking at sort of other assessments that might be useful in this space. And so with that, I want to thank everybody on the team. This is a type of study that requires a very large, very talented team. So a huge thank you to everybody, in particular Jill McKee, who's the PI for this study. And a huge thank you to our families as well. It is a tremendous amount of effort and work to come and see us every 6 months. So none of this would be possible without the huge buy-in from the SYNGAP1 community. So with that, I will take any questions.
Thank you, Alicia. We have time for maybe 2 questions. And I think I stole one of the microphones. I'm going to walk by myself. Yes?
Yep, so
and I can maybe just let me repeat the question. So
Sure.
the question was about the cadence of the study, about whether this is every 6 months or every 12 months.
yeah, so we do ask families to try to come every 6 months if they can, and that's broadly true across the board for the study. But we do recognize that this is the real world, and not every family is going to be able to make it to the clinic absolutely every 6 months exactly. So there is enough flexibility built into the protocol to allow for a little bit of variability in how exactly when families follow up, because otherwise it would be sort of impossible for people to always make that exact mark.
If there are no other questions, we're going to move on to our next presentation. Thank you, Alicia. And the next...